Hi, welcome everyone to Osteobites, and thanks so much for joining us today. My name is Christina Iptoma, and I'm Director of Scientific Programs at MIB Agents and also mom to OsteoAngel Dylan. And today on Osteobites, we are joined by Dr. Brian Ladle from Johns Hopkins University, and he will be discussing deciphering the impact of STING agonists on the tumor microenvironment in osteosarcoma. Thanks so much, Dr. Ladle, for joining us on Osteobites today. Please feel free, everyone, to submit questions during the presentation through the Q&A in Zoom, and we will share those with Dr. Ladle after his presentation. A little bit more about our guest today. Dr. Brian Ladle is a pediatric oncologist, sarcoma specialist, and immunotherapy researcher at Johns Hopkins University, and his research centers around the goal of generating potent immune responses against pediatric sarcoma tumors, especially osteosarcoma. Ongoing projects encompass preclinical work using osteosarcoma mouse models and osteosarcoma clinical trials in both dogs and humans. He really believes immunotherapy approaches can be effective against osteosarcoma and will become a key piece to make it better for osteosarcoma patients. We are thrilled to have you on Osteobites today, Dr. Ladle. Just one announcement before we get started. We are just 4 days away from September, which is Childhood Cancer Awareness Month. And so we hope you go gold with us this September by supporting our Gold Bow campaign. Each gold bow is a powerful symbol of hope, resilience, and solidarity for children and families facing cancer. You can help us turn neighborhoods gold and make a meaningful impact one bow at a time. You can either make a donation to receive one bow, or you can even get a pack of bows, or you can sign up as a bow agent volunteer. And as a bow agent, you'll help raise awareness by distributing beautiful gold bows throughout your community, on mailboxes, storefronts, schools, and more. I just delivered a bunch in my neighborhood earlier this week, and it's so nice to see the neighborhood gleaming gold this September. So I'll put a link in the chat where you can order bows or sign up as a bow agent. And let's go ahead and get started with our presentation today. Dr. Ladle, you can go ahead and start sharing your slides.
Good, thank you. And Christina, I'm thrilled to be here. Right, I've been thrilled to work closely with MIB just the last few years. Right, and I've really enjoyed those interactions. And I keep learning more and more about osteosarcoma through presentations like this, right, and through the work of MIB agents to bring so many camps together to fight this disease. So, all right, great, good. So, so I thought, right, I'd organize this talk in a couple of different ways. One, maybe just to explain, right, why, right, you mentioned in my introduction, right, I do believe that immune therapy can be a key part of osteosarcoma therapy. So why do I believe that, right? And how do we think that can be incorporated? And then go into a couple of specific projects we've been working on in our lab and with our collaborators, looking at specifically this pathway called the STING pathway. And we have some work in both canine osteosarcoma as well as mouse models of osteosarcoma. And I'm glad to kind of take questions through the presentation. I have a set of slides, but if I don't get through them all, no one's hurt by that. But I hope we can have a good discussion and interaction about this topic. So this is the one talk where you don't have to give a slide on osteosarcoma statistics. So just start right into the problem. And so this is I think an interesting graph. This is survival for patients with osteosarcoma through the decades, right? So for instance, starting in the early 1900s through the 1960s, where all we had was surgery alone. You see there was, right, a small group of patients, right, that could do okay with just amputation. But you see is really the advent of chemotherapy as well as kind of supportive care to get patients through chemotherapy that has brought us to kind of where we are today. But unfortunately, or I think is probably all who are joining this call know, we're kind of stuck at this plateau as far as improving outcomes for osteosarcoma just with the therapies that we currently have. And so, right, what are we going to do? How are we going to, kind of break this glass ceiling, right, and really start to make other headways in osteosarcoma? And so I think quite a lot about specifically, as a clinician, I see patients get MAP chemotherapy, and I see a lot of them, that do well, right? We have many long-term survivors that have received just MAP chemotherapy alone. And so it's also as a researcher, you have to kind of take into account that, all right, MAP chemo is not going anywhere anytime soon, and that it is so effective for so many patients, right? It would be very difficult to propose something of, right, we're going to do something completely different in your upfront therapy, right? But are there ways that we can incorporate with MAP chemotherapy, right, to improve its effectiveness? And this is something I think about quite a lot as I think about research as well as clinical trial implications. And right, there's a couple of reasons I think about this. One is that the agents used for MAP, so of course that's methotrexate, cisplatin, right, doxorubicin or adriamycin. There's kind of multiple lines of evidence that how these chemotherapies kill tumor cells can actually be what we call immunogenic, meaning it could actually kind of activate the immune system, how this cell death occurs, right? And is that something that we could potentially use to our advantage? In that, for instance, can we co-opt the immune system to be activated while this chemotherapy is occurring, while the cell death is occurring, as opposed to, right, the typical kind of tissue healing, fibrosis, type response that occurs with cell death? So a second reason, I think, right, is there a reason to think about immunotherapy with chemotherapy, is the agents we use for MAP, they're unique in that, well, not unique, but right, somewhat distinct from many other regimens in that patients, their lymphocytes, these are your T and B cells, those counts are actually very well preserved through standard chemotherapy. And I plotted out just a couple of patients once and kind of measured, right, what is their lymphocyte count throughout therapy? And right, you can see that, right, they start in a normal range and almost end in a normal range. And maybe some of you or your physicians actually don't do, for instance, like Bactrim, these antibiotics to prevent infections that occur with low lymphocyte counts, because really during standard MAP therapy, you have good T cells, good B cell counts that are maintained throughout your chemotherapy. So, right, is there a role then to combine an immunotherapy approach with standard chemo? So this is also, something that has been tried before with other agents. And right, this is a therapy, MTP, right, rafamertide, that is kind of well known to the osteo world, as well as kind of its complications. But this is a potential immune therapy, right? It is a drug that's meant to mimic kind of like bacterial byproducts and things that can kind of non-specifically activate the immune system. And these are studies that have been completed now for some time, but showed these are patients where in these final studies, they were combining MTP with standard chemotherapy. And right, they actually saw kind of an increase in overall survival. These studies, kind of have multiple caveats, which is why the FDA has not necessarily approved this drug here in the United States. But I think many believe, right, that this is an active drug and one that can be combined with chemotherapy. And right, the goal of MTP is an immune activation. So what other evidence exists as far as could the immune system actually potentiate chemotherapy or be helpful during this? During receiving chemotherapy. And this is an interesting paper that was published now a couple years ago by a group at NIH, where they looked at various sarcomas, right, various pediatric tumors, and looked for what % of the cases that they analyzed had significant immune cell infiltrates, right? And that's the axis down here. How many samples that they analyzed actually had significant immune infiltrates. And you see that osteosarcoma, right? It's not all of them, but there's a good number that, for instance, they characterize, and this is via, RNA-seq type of evaluations, that these tumors, a percentage of them had pretty significant immune infiltrates. Now, looking specifically at the osteosarcomas, they saw that the patients, right, the patient samples where they had high, this is kind of a conglomerate immune score, or specifically looking at a CD8 T cell score, those patients who had the highest scores or potential highest immune cell involvement in these tumors had significantly better overall survival. And importantly, these are not patients who received immunotherapy, right? There's pretty much no one gets immunotherapy as a standard for osteosarcoma, but these are patients who received standard care. But if they had more immune cell engagement, more immune cell involvement, they did better. a second group, right, looking at kind of similar phenotypes, and this is a comparative oncology group at the NCI, where they compared both canine osteosarcoma as well as human osteosarcoma and looked for these kind of immune cell signatures that might be present in these tumors. And they classified osteosarcoma in kind of three different classifications based on the amount of immune cell engagement. One, the smallest, right, but it was termed immune enriched. There was a kind of mixed immune enriched, but had a significant extracellular matrix. And then there's a large subset of tumors in osteosarcoma that was termed immune desert, meaning we really just didn't see much immune cell engagement at all. And interestingly, right, both of these phenotypes were present both in the canine data that they looked at as well as the human osteosarcoma data. And when they then stratified overall survival based on these immune phenotypes, you see that both the canine data as well as human data showed that the osteosarcomas that had this immune-enriched phenotype had more immune cell engagement. Again, that resulted in increased overall survival. And again, I'll go back. These are not patients who received immune therapy, right? These were kind of standard therapy that, right, we're able to stratify based on kind of baseline immune cell engagement with these tumors. So, right, in my mind, it kind of leaves this hypothesis that if increased immune cell infiltrates, if that improves outcomes with standard of care therapy, potentially can we improve outcomes with that standard therapy by, right, different ways of bringing in immune cells, right, or engaging these immune cells with osteosarcoma. And right, I might, potentially think of it, there are those immune desert tumors, for instance, and can we convert those immune desert tumors or those ones that are not immune infiltrated, can we convert those to a more immune infiltrated tumor that would then have a better overall survival? So this is right, IHC, this is a slide of osteosarcoma, just to kind of get a little, right, microscopic glimpse into this disease. This happens to be a canine osteosarcoma. And here's the challenge. So this is a stain where we're looking at T cells. So T cells using this method are these small brown cells, right? And you can see in this osteosarcoma sample, right, there's maybe 20, 30, right, just kind of random T cells that are going around. This would be considered kind of like immune desert, meaning there's very few T cells that are engaged in this tumor. As opposed to, right, and this is, I think, an interesting observation. This is not just true for osteosarcoma, but other sarcomas as well. It doesn't mean that the immune system ignores these tumors just because there are no T cells, and that there's definitely a lot of immune cells that are present in these tumors. This is a stain specifically for monocytes and macrophages. So these myeloid immune cells that are present in the tumor. And again, these are marked brown. You can see that they're really kind of all throughout these tumors, right? A significant number of these monocytes and macrophages in the tumor. So that brings us to, right, our kind of forays looking into the C-gas sting pathway in osteosarcoma. And my initial thoughts, going into this were, I know I have all of these myeloid immune cells present in these osteosarcomas. Could I have, right, could there be a drug or something that could activate those immune cells that are already there to, instead of being immune suppressive, be actually immune activating? And that's what kind of got me thinking down this pathway. Right, as of course has become a rather complex story, and as others are also investigating the story, it's not, just kind of it's never a simple solution, but it's one that, for instance, this is our hypothesis, potentially could we manipulate this pathway to induce kind of this immune activation in these tumors that could portend a better outcome. And right, so I'll look kind of at two specific stories that we've been working on in the lab, working on getting published here in the next few months, specifically looking at canines, so some dog osteosarcoma, as well as models of murine, right, or mouse osteosarcoma. So specifically, just a little bit more about what is the STING pathway. So STING pathway is what we term a cell intrinsic kind of danger sensing pathway, and that these are proteins that exist kind of at all times in most cells, that can be activated by different stress or different damage responses. So specifically, DNA damage, viral infection can induce this pathway. And what occurs is that once these products are present in the cell that activates this pathway, it results in activating the protein called STING, which then phosphorylates different transcription factors that then go to the nucleus and are going to induce transcription of various inflammatory cytokines. Okay. What we've been looking at is a STING agonist. And so the STING agonist that we've used is specifically a compound called ADOS-100 and a cousin drug R229 that acts downstream of the C gas. It acts right as a cGAMP analog, which is going to directly activate the STING pathway. And so these are the experiments we've done where we've now been using the STING agonist to, again, activate this pathway, not just in the tumor cells, but also the immune cells that are present. Okay. So is there reason to even look at this pathway in osteosarcoma? We did just some cursory look. This is using the publicly available target data set, simply looking for expression of these different players in the STING pathway, right? And you can see that all of these, right, kind of key players in this activation pathway are expressed in osteosarcoma. As you might imagine, this is RNA sequencing data from a bulk tumor. So of course, right, you're not just sequencing the tumor cells, but also any immune cells that are present. So this data certainly can't distinguish, is the pathway active in the osteosarcoma cells themselves versus the immune cells. But we do see, right, expression of these players in this pathway, right, and would have reason to expect that a STING agonist could activate the STING pathway in these tumors. So I'll first tell you about the project we've been working on in canine osteosarcoma. This is done with the fantastic team of collaborators here at Johns Hopkins, specifically highlight them. Derek Kreichman is a veterinary PhD who her emphasis and expertise is in image and image-guided therapies. So she has actually been kind of one of the instigators of this project as well, in that we're using image-guided techniques to inject the STING agonist directly into the tumor, right? I think that's important, maybe context as far as all the experience we show. We've, right, at least our lab, our technique has been to inject the agonist right into the tumor directly. So after the treatment, our team of pathologists here at Johns Hopkins and comparative pathology have been key to help us understand what's going on in these tumors afterwards. And then all the immune cell, right, and immune analysis that I'll show you has been done by folks in my lab. I'll specifically highlight Michelle Dosset, who has done, right, a lion's share of this analysis and this work. So this is a canine clinical trial. Okay, so these are pet dogs from the community who develop osteosarcoma and are interested in enrolling in a clinical trial. All right, and so they call us and come here to Johns Hopkins, and we treat them. And the dogs actually would go back to their local veterinarian, get the amputation, right, and then finish their standard therapy. So specifically, this is our trial has been limited to dogs with non-metastatic osteosarcoma, so only present in the initial bone. And our study has also been looking at cryotherapy. So this is freezing the tumor. I won't show those results today, right? But I'll specifically focus on the dogs here that we've treated with, again, intratumoral injection of the STING agonist. So this is given before surgery. So the dogs are given a dose 2 weeks before surgery, 1 week before surgery, and then have their amputation. They then go on to receive kind of standard therapy for dogs, which is typically the chemotherapy carboplatin. They're then followed as far as outcomes as well afterwards. But what we focused on is the tumor tissue that we receive, right, on the amputation, that we can then analyze what are the effects of the STING agonist that we've injected into this tumor. All right. So to this point, we've treated 10 dogs. And importantly, as I mentioned, we give it intratumorally via image guidance. So we can see the tumor on imaging. We can see exactly where our needle goes in and where we inject the STING agonist. So that's an important part of our analysis, that we know kind of in that tumor where we injected versus area of the tumor that were not injected with the STING agonist. Are able to compare them. So I think importantly, right, how I think about this, I'm not asking this STING agonist to get rid of this tumor, right? That injection of STING agonist is not going to cure these osteosarcomas. But again, could it activate an immune response in this tumor that the treatment is then amputation, right? That's the kind of primary treatment of that osteosarcoma. So it gives a window into what's going on with the STING agonist injection. Now, importantly, because of the timeline of this experiments, we're not seeing kind of the immediate effects of the injection, but we're seeing it, a week or 2 weeks after the injection. So it's kind of the late effects of the STING agonist treatment that we're observing in these tumors. So how we've done our analysis, we've used various analysis techniques. One of the main ones we've done is specifically called IHC, or immunohistochemistry. So this allows you to label specific different cell types. And in our case, we've been looking at immune cells and 3 different markers for 3 different cells. One is CD20, and these are B cells. CD3 are T cells that are present in the tumor, and then IBA1 is the marker for kind of the monocyte macrophage cells that are present in these tumors. And this is kind of lower magnification, so you see just these little brown dots, that are present. And we specifically compare sites of the tumor that were not injected, so distant from the injection site. Those are the non-STING sites. And then we've compared the immune cell infiltrate to, again, the sites where we know the STING agonist was injected. And I do have to say, this is one thing we've learned in this study, and that the effect of the injection is quite local. That, we see kind of an effect right at the injection site, but, if you go several centimeters away, for instance, the effect of that injection is gone. So it really does seem to be kind of a focal effect where that injection occurs. So just to highlight, this is one example, in the sites that were injected, we saw a significant increase in B cells that are present in these tumors, T cells, where there before just very few scattered throughout. We see large areas of significant T cell and infiltration, as well as often we see also significant increase in these other, the IBA1 cells that are present in these tumors. So as I mentioned, we've treated 10 dogs now, and we've done the analysis on all 10, looking at these tumors and that have been injected, the STING site versus not. So what I'll highlight is that some of these tumors have responded very well, where we see a significant increase. Here is kind of fold induction of the T cells, where if we draw a cutoff of like 2-fold induction, about 8 of the dogs, had a significant increase in that T cell enrichment in the area of the tumor, but importantly, not everyone, Several of the dogs did not see, or had very little infiltration. And so, that's something that we're very interested in. What about the tumors that responded very well, versus those that have not, And these are questions that are important to answer. So looking, that previous slide was T cells. So looking at the IBA1 positive myeloid cell infiltrates, the scale is significantly different, There are a lot more of these cells present at baseline, as well as, some that were significantly enriched. And importantly, when we looked at the ratio of T cells to these myeloid cells, it's commonly thought that the myeloid cells are more suppressive, the T cells are going to be more activating. Then in our STING areas, we see a significant increase in the ratio of T cells to these myeloid cells. And again, I'll highlight, it is not a homogeneous response, There's kind of significant heterogeneity in the type of response, but we are seeing, various tumors where we really are making a significant change in that tumor microenvironment. Looking at the B cells is a similar story, where interestingly, those that enriched for T cells were also those that enriched for B cells, As an immunologist, I definitely see how those would go together. But again, somewhat of a heterogeneous response, but definitely animals, where their tumors did have significant increase in these B cell infiltrates as well. So one limitation of doing studies in dogs is the reagents you have available to study these impacts. Specifically with IHC, there's only a few markers that are used. So we wanted to kind of better understand the infiltrates that are present. And so we used a RNA isolation technique to do that, where we specifically cored areas of the tumor that we knew, again, were not STING injected sites versus areas where we knew there was significant immune cell enrichment in these sites. And again, the question wasn't whether or not there's immune infiltrate, but to try and better phenotype what are the immune cells that are present in these inflamed areas. So taking these cores then, isolated RNA, and use a technology called nanostring to look at RNA expression in these different areas of the tumor. So comparing again a subset of the dogs that had significant infiltration versus those that did not. Right, we saw a significant difference in the differential gene expression, as we would expect, just knowing we had kind of immune cells versus not. But importantly, the questions was what kind of cells or what kind of pathways were upregulated, again, in these areas that were injected with sting. And right, we saw a significant increase in change in pathways such as antigen processing, B cell functions, immune co-stimulatory activation, antigen presentation, T cell function, T cell cytotoxicities. So an increase in all of these pathways, right, that we would expect if there was kind of an immune response that had been generated, right, a robust response that was present there. Now, something that I think is almost just as important and very interesting is what other pathways were induced. So if we got significant immune cell infiltrate there, what else was going on, right, that we need to understand? And this is important in that we also saw induction of various inhibitory pathways, so immune inhibitory pathways. So this is looking at expression of these various, we call these immune checkpoints. These are all proteins that, for instance, have different drugs that could target them. So you see at baseline, they're very low expressed in right, the areas of the tumor without sting present. I'll highlight an immune suppressant cytokine called TGF-beta that is sky high in all of these tumors all the time. That's a well-known feature of osteosarcoma. But specifically in the areas where we saw the significant immune cell infiltrate, we also saw upregulation of these inhibitory pathways, such as the immune checkpoints, right, IDOs, another immune inhibitory pathway. And so in my mind, right, it highlights that, right, sting may be able to increase, right, immune cell infiltrates, but that I would also not expect that maybe as a monotherapy that's sufficient, and that if you can elicit immune response, you then, right, maybe need to come in with, for instance, immune checkpoint inhibitor that would allow that immune response to kind of be released, right, and take full effect. So at least our canine work, right, kind of looking at some of these conclusions, it does seem that the sting pathway is operative, right, in these canine osteosarcoma cells or in the osteosarcoma tumors. I think one thing we cannot understand, right, from our experiments is specifically what's the contribution, for instance, of the tumor cells being activated with the sting pathway versus the immune cells that are already present. The sting agonist given intratumorally can induce a significant activated T and B cell infiltrate. As I somewhat highlighted, right, some tumors respond better than others. That is again, kind of a key question. And it's one reason I've really have liked doing these canine models. These are very heterogeneous tumors, right? It's kind of each one is unique for each dog. And we're able to kind of better look at some of those features of what would be a responder versus a non-responder. An important question is, what is the specificity of these immune cells that are present, right? So we're bringing in T cells and B cells, but are they just kind of passengers, or are they actually there? Are they actually tumor specific, right? This is kind of a key question that we're looking at. And then most importantly is, has this treatment induced a systemic or protective anti-tumor response? That, as I mentioned, the sting agonist is given into the tumor, and then that tumor is amputated, right? That kind of source is now gone. The hope would be that that treatment has generated a systemic immune response, meaning you have T and B cells that are now running through your body that potentially can protect from metastatic disease in the future, right? And this would be a kind of a key goal of a therapy like this. And right, it's a question that we still have in these canine models. And then, as well as I mentioned, the compensatory inhibitory pathways that are also upregulated, right? And can we target these in combination therapies that potentially will work best with sting agonists? And so to kind of transition, this is some of the work that's best done in, for instance, like a mouse model, right? Where we can do, we can look at these different combination therapies, right? What these different pathway questions we can answer very well in mice, right? That are very hard to answer, for instance, in human studies or in canine studies where, right, it's such an investment to, get for each patient and things, right? Whereas the turnaround in the study, we can do a lot quicker in our mouse models. And so I'll transition to talking So about the mouse work that we've been doing recently, I'll highlight Dr. Erin Resch. She's a postdoc, Peds Onc fellow in my lab who conducted most of this work, and specifically looking at some of the mechanisms of immune activation with STING agonists in our osteosarcoma models, as well as what would be potentially different potent combination treatment strategies to go along with it. So I'll highlight the models that we've been using for, again, these are mouse studies. So we have an osteosarcoma model F420 that was generously given by Dr. Jason Eustein, and then sort of a long-time model that's been around, developed by Dr. Chan Khanna, the K7M2, and these are in two different mouse strains, Black 6 and Valve C mice. And again, using the same STING agonist, this ADUS 100. So for instance, when we incubate just tumor cells alone with the STING agonist, we do see some kind of tumor intrinsic activation of the STING pathway, looking at these downstream genes, for instance, interferon beta, CXCL10, CCL5. Interestingly, it seems like the K7M2 model is somewhat more responsive to the STING agonist than the F420 model. But I think that allows us, right, to kind of study maybe a more responsive tumor, right, versus a less responsive tumor. And again, I'll highlight, right, the STING agonist that we're using. This is kind of where it acts in this pathway, right, and that it's going to act kind of directly on STING to induce this downstream induction of these activated genes. So how have we studied this in vivo? And I'll highlight some of the first experiments we've done, and now our questions as far as what's the mechanism that's going on. So as I noted, right, very early in the talk, the potential combination of chemotherapy with a STING agonist, right, or different immune therapy is something that we wanted to investigate. And so in our animal model, what we've done is we've injected these mice with tumor, allowed that tumor to establish, and then we've treated these mice first day, the day before chemo. We give a dose, again, this is intratumoral, so injecting right into the tumor of the STING agonist, followed a day later by a dose of chemotherapy. We started with carboplatin, and as I mentioned, that's kind of most related to our work in the canine model, and we've wanted to kind of model that here. So starting with carboplatin, and then in our in vivo experiments, we've done kind of 2 rounds of treatment, right, similar to how patients would receive very different cycles of chemotherapy. And we've looked at our control where we just inject water into the tumor, the STING agonist alone versus the chemotherapy alone, right, versus the combination where we give STING, then the chemo. So what was very interesting, right, in that the mice that were treated with either STING agonist alone or chemotherapy alone, right, there might have been a little bit delay in tumor, but really no kind of long-term survivors in those experiments. Whereas when we combined both, giving again STING, then the chemotherapy, right, we now saw a subset of mice that were able to completely reject those tumors. And this was true in both the F420 model as well as the K7M2 model, where, again, it was only with the combination therapy that we saw long-term survivors. The K7M2, right, might highlight a little bit more what the individual treatments do, right? STING agonist alone, you'd have some mice that would kind of delay or plateau their tumors. Chemotherapy alone, you'd do the same, but it was really just with the combination therapy that we would get tumors that were present and then would regress and stay away. So we wanted to investigate, right, how is this, right, what are the mechanisms of this? How is this occurring? And so the first question could be, is the STING agonist itself just making the tumor cells more sensitive to the chemotherapy? And this we felt we could model in vitro, and where all you would have is the tumor cells with the chemo and the STING, and looking at their sensitivity to death based on, on the increasing dose of carboplatin, we didn't see any difference as far as adding STING agonist or not in the sensitivity to chemotherapy. So this did not seem to be kind of an intrinsic change in the sensitivity of the cancer cells to the chemotherapy itself. So looking kind of more closely at exactly what was the STING injection, right? How was that changing the tumor? We looked at a different panel of cytokines or immune cell activating proteins that are present in these tumors at different points. So our untreated tumors, tumors that were treated 6 hours before, 24 hours before, 3 days, or a week later, kind of look at what would be the immediate effect versus the late effects. Interestingly, right, with these different cytokines and chemokines, right, kind of grouped. These are kind of myeloid cell trafficking markers, and we saw kind of an acute activation in several of them, or induction in several of them. A little bit of an increase in VEGF, but importantly, as far as pro-inflammatory and activating chemokines that bring in activated T cells and effector cytokines, right, we saw significant induction. Interestingly, it was really quite acute, right? Like 6 hours after the injection is when we saw the peak of many of these, all right? And many of these immediate side effects of the sting injection were gone by 3 days. So for instance, IL-6 was significantly induced, CXCL10, right? TNF alpha, interferon beta was another one. I don't think it's on this graph, but right, we saw significant kind of induction of the sting pathway in these tumors after treating. We then also looked at some immune cell infiltrates in these tumors. This is looking at IHC, specifically looking at T cells. So importantly, the T cell effect was not immediate, right? And that we only saw some difference, right, a week later, and mostly kind of in a subset of tumors, as you might expect with our, not everyone responded, right? But that we saw significant induction in T cells into these tumors a week later. This is looking at some of our other cytokines, right, where we did not see quite as much difference. But interestingly, we also wanted to look at this as far as chemotherapy. Did that chemotherapy itself, did that induce any of these chemokines or cytokines? And the answer was no, right? There was really very little difference in these cytokine or chemokine production with treating with just chemotherapy alone. When we kind of compare, right, chemo-treated versus sting-treated versus the combination treatment, and then looking at induction of these different cytokines grouped together here, right, you really see that the sting signature is what is most overwhelming. So in the combination treatment, whereas given sting then chemo, right, it had mostly the same signature as the sting only treated tumors. And I think, right, interestingly, even though we only saw responses in combination treatment, as far as cytokines go, there was really, no kind of specific signature that occurred with the combination therapy, and that the signature was really kind of sting driven. We then tried to do a deeper dive into the immune cells that were present in these tumors. This was via flow cytometry, so taking the tumors out at different time points and phenotyping the immune cells that are present in these tumors. We did see significant changes in some of the myeloid cell populations, specifically the F480 tumor-associated macrophages. We seem to see an acute drop, again, in either sting or sting plus chemo treated. We saw a significant increase in iNOS-producing TAMs. So iNOS is considered an M1, or kind of an immune-activating effector molecule that's present, right? We saw significant induction there, again, early on. And interestingly, one of the few kind of signatures that we saw in the combination treatment was this early influx in NK cells, right? And this is something we don't fully understand. It's just a very interesting kind of observation that we'll continue to follow up. So looking at the T cells, right, the different T cell populations that were present in these tumors, interestingly, the number of kind of CD4 or CD8 really didn't change that much. But when we looked at the activated immune cells, so the cells that were present that could make cytokine, for instance, CD8s producing interferon gamma or granzyme B, right, that's when, for instance, the later time points is when we seem to see right, an induction in these immune cell, specifically the T cell infiltrates in the tumors. And again, kind of highlighted via IHC here, looking specifically at CD8. So we had a key question, and one of which was, as I mentioned, was it the tumor cells, the sting activation in the tumor cells versus the host immune cells that were really important for this phenotype that we saw. So we have used what are termed golden ticket mice. So these are sting knockout mice. So in this experiment, the mouse itself, right, did not have active sting protein. whereas the tumor cells that we injected into these mice did have active STING pathway. Okay, so our goal was to be able to hopefully tease out what was important as far as tumor cell signaling versus kind of immune cell or host, right, signaling. And what was interesting is in these golden ticket mice, though we saw maybe a little delay in tumor growth, right, we never saw any complete responders or survivors in these combined treatment mice that was only present in the wild type mice, right, meaning this response was really dependent on the host cells, right, as opposed to the tumor cells themselves expressing the STING or being activated by the STING agonist. Trying to investigate a little bit more on the mechanisms. We used a genetically engineered mouse, right, or mutated mouse that's called RAG1 knockout. So these lack both T cells and B cells. And importantly, when we did these treatments, right, again, the combination or individual treatments in the RAG knockout mice, we never saw any complete responders. And so again, pointing to both T, right, and potentially B, we couldn't discern from this experiment, but that these adaptive immune cells, the lymphocytes, are very important for this, for the ability of these tumors to be rejected, right, and to have any long-term survivors. We tried to do a little bit more targeted depletion, where these were wild type mice that we then use antibodies to deplete different immune cell populations. And these were tumors that were established, treatment was started, and then the depletion was started. And so SC, or mice were treated with STING and carbo. You see that mice that were treated with the isotype or control, right? Again, we had this proportion of mice that rejected the tumor. And interestingly, if we depleted CD4, CD8, or a combination, right, we largely got rid of that response. And then of interest as well, we also depleted NK cells, right? And NK cell depletion as well kind of ablated this, this protective response that we could achieve with the combination of STING plus the chemotherapy. So just summarizing some of this data, we've learned that for the combination therapy to work, it requires host STING activity. So I take this as kind of being immune cell or stromal cells present in these tumors that are required. It's interestingly that there's always seems to be this initial delay in tumor growth in all the STING-treated animals, but the tumor clearance, or this ability to actually eradicate the tumor, was completely dependent on either T or B immune cells, CD4, CD8, and NK cells, right? And if we depleted any of those, we lost that ability of the immune system to be able to clear this tumor. So we wanted to focus a little bit more on the macrophage population. That's one of the kind of acute changes that we saw when we gave the STING agonist in the tumor. And so we wanted to investigate this, again, the STING agonist activity on macrophages specifically. So we generated in vitro, so we cultured macrophage populations starting from bone marrow cells and then activated them either with the STING agonist or a well-known kind of immune activator, LPS, lipopolysaccharide. And we saw, for instance, induction both in interferon beta and NOS2, which is, again, the kind of effector molecule macrophages make, we saw significant induction of these in both M1 as well as M2 macrophages. And looking at their activation via flow cytometry, interestingly, they, specifically M1 and M2, both responded to the induction, right, with the STING agonist, right, and that it wasn't dependent on just M1 or M2, but both of those macrophage populations could induce iNOS specifically to the STING agonist. Importantly, we made macrophages from the golden ticket mice. So again, these are the STING deficient mice. So you see that they were still able to skew properly, right, to their different macrophage phenotypes, but did not respond to STING agonists, right? It did not change their phenotype at all. We then did some kind of direct anti-tumor experiments using macrophages plus tumor cells and STING in vitro. So in this experiment, we plated tumor cells that were fluorescent red. So you can then track the growth of those immune cells, right? It's called an incucyte experiment, where the machine actually takes pictures like every hour, every 4 hours, whatever you want to do, of your tumor cells, and you can follow the growth of those tumor cells over time and then do a readout of how many of those tumor cells are present. In this experiment, so we had our tumor cells were red, We then added in macrophages that were either, again, in vitro derived M1 or M2 macrophages, and then plus or minus the STING agonist. So the goal is kind of isolate, again, that effect. It's in a dish. All we have are macrophages, tumor cells, plus or minus STING agonist. It was very interesting, right, that the macrophages themselves had this direct anti-tumor cytotoxic activity when they were given when they were treated with the ADUS 100. And this was STING agonist kind of being given immediately right upon co-culture. And right, that just macrophages themselves, right, did not have anything. STING agonist itself, right, did not change the effect, but it's really the STING plus macrophages, right, were able to kill these tumor cells, which I think is very interesting and in work that we're pursuing more. So could this explain kind of this initial tumor growth that we see in all these STING-treated animals? Could this kind of acute immediate effect be more macrophage dependent, whereas tumor clearance is then dependent on the ability of the immune system to mount that adaptive immune response, right, and recruit in these T cells, and other adaptive immune cells? So key questions from this data and things that we need to understand better and want to pursue more. For me, the question of responder versus non-responder, we talked about this a little in the canines, but it's even more intriguing in these mice where these are genetically identical mice. These are genetically identical tumors, and you have some respond and some don't. Right? What is going on in kind of the tumor microenvironment, right, that allows almost that, is there a balance or something that tips, right, to allow kind of an immune response or eradication versus those that don't? This effect where, we see this response in some mice, but not all mice, definitely begs other combination therapies. So what else could we add to this to, again, improve its efficacy? The experiments I did that I showed were using the chemotherapy carboplatin. Is this effect specific to that drug, right? Or other chemotherapy drugs, will they be synergistic as well to add, right? This is a key question. And one that we're asking specifically with the drugs we use, right, in people up front. So the doxorubicin, right, carbo and cisplatin are cousin drugs, but methotrexate as well. We specifically are investigating whether immune checkpoint inhibitors, so again, this would potentially release or allow a T cell response to be more active. And some of our initial experiments are that we're seeing much better responses if we add an immune checkpoint inhibitor as well as the STING agonist and chemotherapy, right? So can we bring up that responsiveness significantly? Are there other adoptive cell therapies that could go very well with this? I mentioned it seems like the NK cells are potentially important, right? Could this be kind of a key way potentially to use NK cells or other immune cells and allow them potentially to traffic better to these tumors, as well as other modifiers of the tumor microenvironment, right? I mentioned earlier TGF-beta. I think this could be a key pathway that can be targeted now with drugs that are already in clinical trial, and could they combine well? I think an important question is the delivery of the STING agonist. Others, have studied, for instance, systemic STING agonist, where you give it IV, for instance. We've been doing intratumoral. Is that key? Is that important? I think these are all questions that we're trying to understand. And then importantly, would an approach like this protect against metastatic disease? And that, as we know, for osteosarcoma patients, this is a key. An effective therapy has to be able to protect against metastatic disease, either treat or potentially prevent. We've done now some initial experiments, for instance, in our mice that rejected the tumor. When we re-challenged with tumor, those mice were still protected, right? So we do think there's evidence that potentially this treatment is generating some immune memory, right, against these osteosarcomas. So just in conclusion, just kind of go over perhaps our model, right? We initially start with a tumor where there's tumor cells, as well as kind of immune suppressive, largely myeloid cells that are present in these tumors. We give the STING agonist injection. We think there's probably some initial cell death that occurs, as well as some transient depletion of those macrophages that then potentially allow the generation of a systemic immune response, right? And then later in immune cell infiltration into these tumors, not just right now with kind of T and B cells and other immune cells. right? And could this combination, again, with chemotherapy, result in this improved tumor cell death? So maybe just kind of going back to the beginning, right? Why would I be interested or excited about combining immune therapy with chemotherapy? I think for osteosarcoma, what would successful immune therapy look like? I think if we can learn how to combine it with chemo, there's great advantages there. And like I alluded to, potentially that could be beneficial to the immune therapy. We have to be able to address the immune suppressive and tumor microenvironment that's present. Are there also kind of systemic immune suppression, right, that's occurring because of the tumor, because of these immune suppressive cells that are there that perhaps we have to adjust, right, or we have to address? I am a firm believer that it's going to require combination therapy approaches. I don't think a STING agonist by itself is going to cure osteosarcoma. And I mean, I say that and maybe it sounds obvious, but for instance, working with drug companies or designing a clinical trial, right, you have to be able to, I think, realize, right, what are we expecting? What could we expect with this, as well as not give up when, for instance, a monotherapy itself does not work. And I think this is key for immune therapy approaches, that it's going to require combination therapies. I think our data is very interesting to just show that it's important and it's beneficial to kind of engage all aspects of the immune response, right? Both myeloid cells, T cells, and B cells, and that, engaging all of those, I think, is going to give our best outcomes, as well as to defining, right, are there specific immune targets, right, or what would be the best? Are there multiple immune targets that we can go after as well? So I'll end there and just acknowledge those that have done the work and our funding. I tried to highlight throughout the talk, but specifically with our canine work, right? Dr. Kreichman, Dr. Brayton, Dr. Gabrielson are key collaborators there, and then members of my lab, right? There's multiple other projects, but kind of the key ones presented today: Michelle Dosay with the canine work, and Aaron Resch with the mouse and STING agonist and chemotherapy. So thank you very much. I'd be glad to take questions for a few minutes.
Hey, thanks so much, Dr. Ladle. And I have to say, every time you said golden ticket mice, it made me smile. So whoever
I don't know who came up with that.
Yeah. We have a couple questions that came in, so let's start with those. So first question, given the highly localized effective, and you did start to touch on these, and you kind of just raised a lot of interesting kind of discussion questions that do need to kind of be examined further. But given the highly localized effective intratumoral STING agonist delivery, how do you think we might leverage STING agonists for the high proportion of patients who develop metastasis, especially given the toxicity of systemically administered STING agonists and the divergent expression of STING across human tumor samples?
Mm-hmm. Yeah, right, it's a key question that's key for, right, almost every immune therapy, right, as well, right? How are we going to get these where we want them, right, in a way that's going to treat the tumor but not cause too many problems? I do think that intratumoral delivery, it's especially in somewhat of a rare population like osteosarcoma, it's certainly feasible. There are right, even for instance, metastatic deposits like lung and things, right? These are sites that could be, right, that could be approached right, intratumorally. Our interventional radiologists are kind of very good at what they do, right, as far as that ability. It's a key question, one that we haven't finalized yet. Obviously, is it required to actually, right? Is intratumoral, does that work better than systemic? Right, that's something that we don't know. The other question about systemic effects of the STING agonist, I think, is also an important one, right? And how do we kind of balance those two? There are new STING agonists being developed as well that kind of seem to do better as far as kind of the systemic toxicity of those STING agonists. So I do think that obviously highlights some of the difficulty with this, but also I think there's ways around kind of all those roadblocks.
Yeah, just to follow up on that. So, because I know intratumoral, like from a patient perspective, that always raises a few eyebrows just because of the, difficulty. So, yeah, interested in kind of systemic delivery, similar to like something like MTPPE, right? Which is
Mhm.
delivered systemically. But I'm also
Yeah,
wondering,
yeah.
because, in your canine studies, because in dogs, they amputate, like you amputate kind of right immediately, right? Whereas in humans, it's like we have 2 cycles of
Yep.
chemotherapy prior. It'd be interesting to see, like, if there's further effect because the tumor is
Mhm.
there longer.
Yeah, yeah. The ability to study this in clinical trial, right, is there's a lot of kind of obstacles to it, but I mean, I really think this could be a therapy you could incorporate into upfront therapy, right? Right now, we're not doing anything in that space, right? We're adding in cabozantinib, but for instance, intratumoral injection into a large primary osteo is not as hard. Um, these are patients who are all going to surgery, right? So, for instance, you'd also be able to learn a lot in those patients, right, who are being treated. So it wouldn't necessarily have to be a study that needs 500 patients, right? We'd be able to, I think, learn effects of the STING agonist, right, from a much smaller cohort of patients. Of course, you have the tumor necrosis, right, that's occurring from chemo, right, all those things. But as we learn kind of what an immune signature looks like of the STING agonist, right, that we would be able to, I think, be able to decipher pretty easily, right, once we kind of have characterized what that signature is. So I do think that opens up, clinical trial opportunities, for instance, in, even upfront patients, right, in earlier stages.
Great. Another question that came in. So, based on the in vitro elimination of murine OS cells by co-culture with the M1 and M2 macrophages plus a STING agonist, why do you think this response was not observed in vivo? Depleting T and B cell populations ablated tumor elimination. So, you believe that this TAM cytotoxicity is suppressed or absent in vivo?
So when I didn't show some of this data, so for instance, when we do in vivo, right, when we inject when we inject STING agonist, for instance, we looked at, for instance, cleave caspase 3. And that's a marker of apoptosis, right, of dying cells. We did see kind of an immediate, within 24 hours, right, increase in cell death just in the tumor. So I do think it is occurring in vivo. We often see kind of this little bit of a delay, right, in tumor growth, and I do think that's occurring, right, in these animals. But I think that response has to be coupled with converting to kind of an adaptive immune response, recruiting in your T and B cells, that's really required to kind of eradicate these tumors. So we do see evidence kind of the macrophage killing in vivo as well, but I think it requires kind of all aspects of the immune system.
Okay, another question. Have you considered combining an additional immunotherapy such as anti-PD-1 based on the successful immune activation generated by the STING agonist?
Yeah, so the in the canine, right, that's one thing we would really like to do, and that there is now an anti-PD-1 or antibody that's approved for canines. We're working on trying to get access to that if anyone has connections. But yes, absolutely right. I do think it's going to take a combination therapy to improve it. What we've now done in the murine models, right, we've combined it with anti-PD-1 and anti-CTLA-4 antibodies in the mice, right, and are seeing much better tumor rejection than we are with kind of the STING and chemo alone. So yeah, there's I think kind of multiple pathways that right, we're interested in. And right. I feel a little bit, sometimes I'll use this analogy like starting a fire, right? If there's no fire, if there's nothing going on, if you pour lighter fluid on that wood, nothing's going to happen. So for instance, could a STING agonist or molecules like that, could they kind of spark an immune response, right? Get something started and then, add immune checkpoint inhibitor, right? Add these other drugs that can do something once you have an immune response started. But if there's nothing going, right, and we've shown this with osteosarcoma, if you just add immune checkpoint inhibitors with osteo, right, very, very, almost no responses, right, that we've seen. But if we generate a response, then use immune checkpoint inhibitor, for instance, right, can we now augment that, right, and make it more of a, successful immune therapy?
Okay, we're pretty much out of time, but I wanted to just kind of one other question, just because you did touch on this a few times. And I was also kind of reading, I think, a poster you had presented somewhere recently about, I think in the Canine study, like the importance of sequencing. And so just kind of the question of frequency and sequencing. And so kind of that idea of persistence. I know you mentioned, I think there was one point where you said you saw like kind of the effect lasted for about 3 days or something like that. But
Mhm,
kind of,
mhm.
so I guess kind of what that tells you in terms of implications for frequency and sequencing, and then especially in terms of clinically translating to combining it early on with MAP and
Yeah.
kind of sequencing it with MAP.
Yeah, yeah. I especially kind of in the sting agonist, right? In that literature, there does seem to be a difference in like a pulse, right? Like there's kind of a pulse response versus a tonic signaling, right? And that if you have kind of tonic activation of the sting pathway, I don't think that's necessarily good, right? There's some reports that could actually induce more metastasis, right? It's kind of a mixed bag understanding this pathway. But for instance, in this setting where it is kind of a one injection, right, or maybe every 2 weeks, something like that, right, where you get this pulse of inflammation, this pulse of immune activation that can occur, right? I think that's going to be kind of the most useful application for it. And I think sequencing with chemotherapy, right, is a interesting question. I'm always struck by, for instance, in the lung cancer world or something, they just say, no, we're going to do it. We're just going to go for it, right? They did immune checkpoint inhibitor with chemo. They just threw it in there, right? And they see all sorts of, they see much better responses, and those are things people never expected, that immune therapy with chemo, that doesn't make any sense, right? You think chemo is killing your immune system. But we're learning that's not true, and it's not true for all drugs, right? And I think there's windows there, right? There's opportunities there that we can learn, I think, applied osteosarcoma.
Yeah, thanks. That was kind of like a myth buster for me when you started off with that, because again, normally you're thinking like, wait, I'm getting this chemotherapy, it's depleting my immune, system.
Yeah,
How
yeah.
is it going to work with this? So, but I think your explanation in the first half really kind of set the scene well for that. So, thank you so much, Dr. Ladle, for joining us on Osteobites today and for making it better for osteosarcoma patients. More information on this and all Osteobites can be found on our website at mibagents.org, on the MIB Agents YouTube channel, and at your favorite podcast place. We hope you can join us on Osteobites on September 11th. We are going to be joined by Dr. Yanding Zhao from the Christina Curtis Lab at Stanford University, and he's going to be discussing how distinct patterns of chromosomal instability fuel osteosarcoma progression and influence patient outcomes. You can find our Osteobites lineup for the next few months on our website. Also, you can go to our YouTube channel and subscribe to make sure you never miss an episode. And if you have any ideas for future topics that you'd like to hear about, please email us at events@mibagents.org. Thank you again so much, Dr. Ladle, for spending time with us today. And thanks to all of you for taking out of time out of your day to join us. And we hope to see you back here in a couple weeks when we chat with Dr. Yanding Zhao from Stanford on September 11th. Thank you. Thanks, Dr. Ladle. Thanks, everyone.
Thanks again. Thank you.